a new couple of seminars. We have Davide Carbone today and Benedetta Mariani. We start with Benedetta, which I briefly introduce now. Benedetta Mariani, she got her master degree in Fisica dei Sistemi Complessi in Torino. Then she earned the PhD in Neurosciences at the Neuroscience Center in Padova, in collaboration with the Laboratory of Interdisciplinary Physics, also in Padova. And this happened very, very recently in January 2025. Now she is starting her postdoc in these two institutions in Padova. The title of the talk today will be Spontaneous Oscillations and Avalanches are linked to whisker sensory response processing in the rat thalamocortical circuit. Benedetta, you can start sharing. Thank you.
The screen.
You have some 25 minutes and then some questions.
Can you see my screen?
Yes.
Okay. Thank you for the invitation and for the introduction. As you said, I will talk about oscillations and avalanches in the rat barrel cortex. So, the motivation of this study is the fact that, as already some talks in this series of seminars have said, is that the cerebral cortex is often in a restless state of activity, even in the absence of external stimuli or tasks. Indeed, it is known and it is peculiar that the spontaneous activity consumes about the 20% of the metabolic energy consumed by the brain. So it is reasonable to think that this spontaneous activity has to be functional, and it is not just noise, as some works the past years have thought. So the main question of the work that I will present is whether this spontaneous activity is in some sense related to task-evoked activity. So two main collective behavior are often observed in neuronal activity. The first is neuronal avalanches, and they were observed for the first time in 2003 by Becks and Plants, that observed neuronal avalanches in rat cortex slices, and they measured the local field potentials on an array on these slices, and were able to detect events of activity after thresholding the signal. Once thresholded the signals, they were able indeed to detect these events of activity, and they were able to group them together into avalanches and to resolve this spatiotemporal organization of neuronal activity. In particular, they found indeed that these avalanches had particular distributions. Indeed, they were power law distributed as regards the sizes and the durations. And they linked these distributions to the criticality hypothesis, in the sense that there is the hypothesis that the brain might operate at the critical point of phase transition. In any case, these avalanches reveal a non-trivial organization of the spatiotemporal structure of neuronal activity. Another collective behavior in the neural activity that is often observed are oscillations. Indeed, it is known that several rhythms have been observed in neuronal activity, and the first of which was the alpha wave that was observed in the electroencephalogram. Or whether they're functional, and in particular in the last year, thanks to modern technologies, we are able to prove whether these rhythms are functional or not, since we are able to actively manipulate some cortical regions and see whether these oscillations are still present or not. So there is still the idea that these oscillations have a functional role, also because they occur often in concomitance with stimuli, sensory stimuli. Another aspect that is important from the point of view of the functional role for oscillation is the coupling between different rhythms. Indeed, as in this picture, I am showing a phase amplitude coupling between two different rhythms, between a theta that is a slow one and a gamma that is instead a faster one. Indeed, the gamma cycle is a very fast rhythm that will be very localized in the cortex. Instead, thanks to this coupling, the phase amplitude coupling is able to transmit possible information contained in these cycles to distal areas in the cortex. So the system on which I will study this collective behavior is the barrel cortex of rats, and it is a controlled system, and indeed it receives tactile inputs from the whisker of the rats, and the whisker of the rats are actually a very important sense for them. Indeed, rats are nocturnal animals, and they use the whisker as we use our hands basically to explore the environment. And the question that I will pose is which is the interplay between the spontaneous and the evoked collective behavior in the rat barrel cortex. And as this picture is showing, I will both analyze some mesoscopic activity that is measured by local field potentials data, as shown here, and also some neural activity that includes also single spikes that is called multi-units activity. So in the first part of the talk, I will present some results that we have published on the neuronal avalanches in the rat barrel cortex. And indeed, we have studied neuronal avalanches in the rat barrel cortex, both during spontaneous activity and after the stimulation of the whisker, after a controlled whisker stimulation. And what we find is that during spontaneous activity, we found some power law distributions in avalanche sizes and lifetimes. Moreover, we were also able to see that these distributions respected some relationship between the exponents that are expected to hold at criticality. On the other hand, we measured the same aspect also after whisker stimulation, and we see that the first part of the distribution is preserved, and also in this case, the distribution follows a power law, with the difference that there are some bumps in the distribution, and these bumps highlight the presence of an excess of avalanches with large sizes in correspondence of an external input. And indeed, if we scrutinize better the neuronal activity underlying these avalanches, we see that after the stimulation of the whisker, there are synchronous activations of a pattern that give rise to these large avalanches that we see in the distribution, and that already at this stage pointed to some coherent organization and activation after whisker stimulation. Indeed, in the second part of the talk, I will analyze both oscillations and avalanches in the rat barrel thalamus network. Indeed, the thalamus is a crucial part actually of the circuit that encodes stimuli from the whiskers of the rat. Indeed, at the level of the barrel, there is a subdivision into columns that have a direct correspondence with the whiskers on the snout of the rat. And this correspondence can be already seen actually at the level of the thalamus, in which we have some subdivision called the barreloids. And we are able to measure and study the activity of both the barrel and of the thalamus thanks to a neural probe that covers both the barrel cortex and the thalamus simultaneously. So we first performed oscillations analysis after the whisker stimulation by averaging the activity over all the repetition of the experiment that we have. And we can see that after the stimulation of the whisker, there is the emergence of an oscillations with a frequency close to 11 Hz in the barrel cortex, while we don't see these oscillations in the thalamus. The same aspect is evident when we analyze local field potentials that, as I said, are a more mesoscopic measure of neuronal activity, and we see indeed this activation at 11 Hz after whisker stimulation. Moreover, we can notice that there is a slow rhythm with frequency below 4 Hz that seems to be present instead during the whole recording. So the aim was to analyze these spontaneous oscillations also during spontaneous activity, indeed, not only after whisker stimulation. And here I briefly mention a technical detail in the sense that we use empirical mode decomposition to analyze the frequency content of the signal that allows us to decompose the signal into modes with well-separated frequency content and track the frequency as it evolves in time, also during spontaneous activity. So we wanted to analyze the frequency close to 11 Hz, also during spontaneous activity in the barrel and in the thalamus. And what it is evident is that in the barrel cortex during spontaneous activity, there is still these oscillations, and that is much more present in the barrel with respect to the thalamus. Indeed, if we perform some statistical test, there is a significant difference. We see that there is the mode with frequency below 4 Hz that is present during the whole recording. And moreover, we can study indeed whether these two rhythms have a relationship with one another, and we can see that they are nested in the sense that the phase of the low frequency oscillation modulates the amplitude of the higher frequency oscillation, revealing a sinusoidal relationship that is significantly different from a random realization. As I said, as another aspect that I can study during spontaneous activity is the organization of the neuronal activity into neuronal avalanches. And I'm also in this data set, I am able to see that the neuronal activity is organized into neuronal avalanches that are power law distributed. Moreover, we can link these neuronal avalanches to oscillations. Indeed, we can see that as before, the slow oscillation was modulating the amplitude of the higher oscillation. Also, in this case, we have that the phase of the slow oscillation modulates the fraction of avalanches. That is a measure of occurrence of avalanches in a given fraction of time. And as can be seen in the figure below, the fraction of avalanche tends to be higher at some precise phases of the slow oscillation. So going back to a comparison between evoked and spontaneous activity, and by comparing the differences in the power of these two different rhythms, we can say that the 11 Hz mode is significantly amplified after the whisker stimulation in the barrel cortex, yet it is still present during spontaneous activity. On the other hand, the mode with frequency below 4 Hz is present during overall the recording, and there are no significant differences, and we can link it to anesthesia or even to respiration. So in order to better understand these aspects, to understand how this phenomenology arises, we develop a model. In particular, the model that we consider is grounded on the literature. Indeed, it is based on a model that was developed by Pinto et al. In 1996. That is a reduced model that was derived from a detailed model instead. The detailed model consisted in 100 equations for the cortex, for the barrel cortex circuits, and thanks to analytical reduction techniques, it is possible to reduce this detailed model to two equations that describe the activity of the barrel cortex. It is important to notice that the detailed model was fitted on empirical data of the barrel cortex responses to some microscopy study. So the parameters that are fitted in the models are biologically relevant. And the procedure that consists in deriving the reduced model just consists in performing reduction techniques that finally gives two equations, as I was saying, for the barrel cortex, one for an inhibitory population and one for an excitatory population, and one equation for the thalamic activity. And the final equations we end up with are very similar to the ones of the Wilson-Cowan model that is a celebrated model in neuroscience. So as I was saying, these are the final equations we end up with. And these quantities on the right-hand side is the firing rate. That is the one that we can actually compare with experimental data. And in particular, it is important to notice that we give as input to the model the experimental firing rate of the thalamus that is here indicated by this square. So the experimental firing rate of the thalamus is the input of our model. And we took the same parameters as the authors of the original paper, and we only varied these parameters, however, by keeping it within the standard deviation provided by the authors. So we varied these parameters that links the thalamic external population with the inhibitory population of the barrel cortex. And by decreasing this value, we were actually able to see a limit cycle in the cortical circuits with the frequencies between 11 and 15 Hz. So in remarkable agreement with the ones that we observed in the experiments. So these are the results of the simulation of the model by giving as inputs the experimental firing rate of the thalamus. In particular, on the left, we have an example of spontaneous activity, and it is possible to see that every time that the thalamic activity overcomes some threshold, the cortical activity exhibits oscillations in a range close to 15 Hz. And indeed, the same thing is happening when we consider the thalamic firing rate after the whisker stimulation. Indeed, consistently over all the trial, after the whisker stimulation, we have an increase in the thalamic firing rate, and thus the cortical model displays oscillations. This same aspect is evident if we study the decomposition into frequency of this model. Indeed, we can see that also at spontaneous activity, we have this frequency between 11 and 15 Hz in the model that are amplified in the model considering the activity after whisker stimulation. The slow rhythm instead is of thalamic origin, and it is due to the thalamic firing rate that we give as input to the model. And we can study also in this case whether these two rhythms have a relationship with one another. And indeed, also in this case, we see that the phase of the slow oscillation modulates the amplitude of the high frequency oscillation, reproducing the relationship that we see in the experiment. So this model is able also to reproduce this phenomenology of the nesting between frequencies. In particular, we can also study a measure of fraction of avalanches. Indeed, the firing rate is a measure that is very similar to the measure of fraction of avalanches, and study the relation between the phase of the slow oscillation and this measure. And also in this case, indeed, we can see that even in the model, the phase of the slow oscillation modulates the fraction of avalanches, and there is a preferred phase for the occurrence of avalanches, as also in this figure on the right, it is possible to see. Then it is important to notice that in this model that we have just described, we have basically a self-sustained oscillator. Indeed, we have a cortical circuit that displays a limit cycle. And so we have a self-sustained oscillator that does, from the prediction of nonlinear dynamics theory, should be able to synchronize with an external stimulation with a given frequency. In particular, it should be able to synchronize even though the frequency of stimulation is different from the intrinsic frequency of the oscillator. And in particular, as the Arnold Tongue predicts, if the amplitude of the external stimulation increases, also the plateau of synchronization of the oscillator should increase, and the oscillator should be able to synchronize also with frequency that are far away from its intrinsic frequency. And this is what we observe indeed by simulating our model and by giving the input some sinusoidal inputs with different frequencies and different amplitude, so we can obtain the Arnold Tongue in our model. And we can also study the phase response curve of our model. That is just another way in which to see the Arnold Tongue. Indeed, the phase response curve is obtained by simulating the oscillations of the models at the different phases and by seeing the phase shift that the simulation induces in the model. And a phase response curve of this kind means that the oscillator is able to synchronize both with frequencies that are in advance with respect to its own rhythm, and both with frequencies that are in delay. So this is the prediction of our model, so that these should display this Arnold Tongue, and we are able to test this hypothesis with further experiments in which we are able to deliver to the barrel cortex some train of stimulations by stimulating, in this case, directly the cortex with simulating directly an electrode of the neural probe. So we are able to deliver some stimulations with a different frequency, and we can show that the neuronal activity of the barrel cortex entrains to this different stimulation frequency. Moreover, indeed, we are also able to build the Arnold Tongue from the data by varying the amplitude of the injected current that stimulates the data. And we are able thus to reproduce the fact that the synchronization plateau increases with the increase of the amplitude of the stimulation frequency by reproducing that the prediction of our model that thus seems to be a good descriptor for the barrel cortex circuit. So I am coming to the conclusions. So we have studied the thalamocortical circuit, and we observed that already during spontaneous activity there are oscillations with frequency close to 11 Hz, and as low oscillations are present. And we find also neuronal avalanches during spontaneous activity in the barrel. We noticed that 11 Hz oscillations are the ones that are amplified after stimuli. And we study a detailed thalamocortical model that explains the formations of 11 Hz oscillations thanks to the slowly oscillating thalamic input. And the slow oscillation modulates avalanches in the model and reproduces the phase amplitude coupling that we have seen also in the experiments. Importantly, the model predicts the oscillatory response to the stimulation that we see in the experiment, and it reproduces the Arnold Tongue, and that we are able to test with a further experiment by stimulating the barrel cortex with different train of stimulations with different frequencies. So by concluding, I wanted to say that the 11 Hz oscillation is actually close to the frequency of whisker twitching, that is the frequency of the muscle contraction of the rodents when they explore the environment, indeed. And this is called whisker twitching. Thus, it is remarkably close to this frequency that is between 7 and 12 Hz. So we believe that this is just not a coincidence, and indeed that they might be linked, and that the barrel cortex could behave as an oscillator with this characteristic frequency, and that this would allow a flexible synchronization between different cortical regions in order to perceive the stimuli. And it is relevant that we already observed this feature during a spontaneous activity that is characterized by a rich dynamical state. So I have come to the conclusion of my talk. I thank you for your attention, and I thank the collaborators of this work and the group of the Laboratory of Interdisciplinary Physics, in particular the neuro part, that have helped me a lot with the discussions to develop this project.
Okay. Thank you very much, Benedetta, also for being perfectly on time. So let's see if there is any questions. I don't see any hand raised. So maybe I can ask one question regarding this idea that this 11 Earth frequency is maybe triggered by the, let's say, natural frequency of the whiskers. Are there any similar experiments in different animals where maybe you can see different frequencies, or this is the only one?
Okay. Especially in rats, there are some experiments in which during, for example, indeed exploration of the environment, they have seen some similar frequencies to the one that we find, and also some other works reported some higher frequency, higher than 15 Hertz. However, it is some sort of an open question because it is still not indeed solved in the literature whether the barrel cortex displays this kind of oscillations. So there has been some reports similar to ours, but it is still an open line of research.
Okay, thank you. Any other question? Okay, if not, so let's thank Benedetta again.